Good afternoon, everybody. My name is Sonoko Ogawa from University of Tsukuba. I'm very glad to have a chance to talk to you about our recent work on social behavior. My talk title today is Lifelong Action of Steroid Hormones on Neural Networks of Social Behavior. Before my scientific talk, I would like to introduce our university and our new neuroscience graduate program. City of Tsukuba is located northeast of Tokyo. From center of Tokyo, called Akihabara, it is only 45 minutes by train to the center of Tsukuba City. This Tsukuba City is bottom of Mount Tsukuba, and it is called Tsukuba Science City. This is a little bit old data, but there are more than 300 research institutes and over 20,000 scientists. Since Tsukuba City is about 50 kilometers from the center of Tokyo, we can enjoy cherry blossom in spring and a lot of flower during summer and foliage in the autumn. We also have some nice lighting of Christmas during winter time. Our university is located center of this Tsukuba Science City like this. It is one of the biggest university as land-wise in Japan. Starting this April, we started a new interdisciplinary neuroscience program here in Tsukuba University. Faculty members from human sciences, such as behavioral neuroscience, behavioral neuroendocrinology, and so on, and faculty of medicine, such as cellular and molecular neuroscience to all the way to systems neuroscience, join this program. Although our original research buildings are very far from each other, we all have a research lab now in Research Building D in this part of the university. We have 4 major areas of neuroscience, such as cellular molecular neuroscience, systems neuroscience, cognitive and behavioral neuroscience, and also clinical and translational neuroscience. The faculty members from both human sciences and medicine together teach all of these 4 major parts of neuroscience. Therefore, this is a very interdisciplinary program in neuroscience. Now, too much delay, I would like to start my scientific part of the talk. As my talk title says, I'm interested in social behavior. Throughout our life, we are living to create and maintain social relationship with others. As Maslow's hierarchy of needs says, being social is a very important part of our life, especially now under the circumstance of spread of COVID-19, which forced us to stay home and keep social distance. We really know the importance of being social. My research focus is to understand how we establish and maintain various social relationships throughout prenatal to all the way to old age. Particularly, we want to know how the hormone act on the brain to regulate various social behavior. This type of research field is called behavioral neuroendocrinology, which is part of behavior neuroscience. Since I heard many of you are from undergraduate schools in various different fields, I would like to introduce some of the basic knowledge of behavioral neuroendocrinology. In this lecture, I don't want to go into too much of detail of neuroendocrine regulation, but in our body, there are a number of major endocrine glands, as shown here. Production of and secretion of hormones from these endocrine cells are regulated by signal from hypothalamus through pituitary. Hormones secreted from these endocrine cells have some effect on target cell to produce biological response, and at the same time send a negative feedback to pituitary or hypothalamus so that secretion of hormones are shut down to a certain point. However, this so-called neuroendocrine system is not the topic of today's lecture. In today's lecture, we focus on these two groups of steroid hormones, which is secreted either testes or ovaries. Testosterone, which is part of androgen, is mainly secreted from testes. On the other hand, estradiol, which is major class of estrogen, secreted from ovaries. Most important concept of today's lecture is these steroid hormones actually are transported through the bloodstream all the way to the brain. There, these steroid hormones regulate neuronal activity in the brain area involved in various behavior, including social behavior. In other words, these steroid hormones secreted from gonads can have central action to regulate behavior. This is the focus of today's lecture. In the previous slides, I said hormones secreted from certain endocrine cells can be transported to the brain. This is the basics of how hormones works. So, certain hormones are produced and secreted from specific endocrine cells. Once it's secreted, go into the bloodstream and finally binds to the target cells, which has specific receptor for this hormone to bind. Each target cell has their own characteristics. Therefore, hormone can produce specific physiological effect on target cells. This kind of endocrine communication is very different from neuronal communication through synaptic transmission. Information only transmitted to presynaptic neuron to postsynaptic neuron, which has by way of neurotransmitter secretion in the synaptic cleft. In other words, in this case, already established neuronal circuitry is necessary to transmit information. This is very different from this endocrine communication. There are three types of hormones based on their chemical structures. One is peptide hormones, consist of this amino acid. The other major class of hormones are steroid hormones, which is basically the lipid. Estradiol secreted from ovaries and testosterone secreted from testes are both steroid hormones. The other important point is mechanism of action are very different between peptide hormones and steroid hormones. Peptide hormones are basically protein, therefore it cannot go through this lipid bilayer of the cells, including neurons. So it has to bind to the receptor which is open outside the cell. Once peptide hormone binds to this receptor, G protein system is kicked in, and through second messenger system, the number of biological function is manifested. On the other hand, steroid hormones are made of cholesterol, and it can go through the lipid bilayer of the cell membrane. Once steroid hormone reaches to the cytoplasm, the steroid hormone binds to their own specific steroid hormone receptors, and this complex hormone receptor complex moves to the nucleus of the cell and binds to the certain part of the DNA to regulate transcription of various genes located in downstream that eventually produce new proteins such as peptide hormones, enzymes, receptors, growth hormone, etc., which are all very important and involved in the regulation of behavior. This steroid hormone action mediated by DNA is called genomic action. Now, in this section, I will talk about how steroid hormones regulate sexually dimorphic expression of social behavior. As I show in this slide, hormonal actions which regulate these various biological events is also very important for the regulation of social behavior. Also, these events are basically very similar between humans as mammals and mice and rats, which I study. Particularly during perinatal period, which right before the birth and right after the birth, testes secrete high level of androgens such as testosterone. However, right after the birth, the testosterone levels became very low and increase again starting at puberty. In contrast to testes, ovaries are very inactive during this period and do not secrete any estradiol until time of sexual maturity. It is very important to understand steroid hormone has two different actions, which is organizational and activational actions. Both of them are necessary to produce any sexual difference in the behavior. As an organizational action, steroid hormone act on the central nervous system to build sex-specific or sex-typical neural structure of various behavior, including social behavior. On the other hand, during adulthood, steroid hormone acts on the neuronal structure which is built by this organizational action and regulate expression of behavior. During early stage of development, there is two important. I want to introduce two difference between sexual determination and sexual differentiation. Today's talk is about sexual differentiation. On the other hand, sex determination is when things happen. One is sexual determination, which is determined by SRY gene, which is located in Y chromosome of sex chromosome. Therefore, if individual has Y chromosome, SRY gene is expressed and develop testes, which secrete androgens perinatal period. On the other hand, in XX individuals, due to a lack of SRY gene, testes are not developed and rather ovaries are developed. Therefore, no androgen secretion during perinatal period. These individuals become females, and in mammals, the individual which has XX is a default setting. On the other hand, sexual differentiation is a different concept. In this case, individual develops either male type or female type characteristics in terms of both physical and behavior. To summarize these things is, at the beginning of development, not only gonad but also brains are undifferentiated. Individual which has XX sex chromosome develops male type brain. On the other hand, individual which has XY sex chromosome develop male type brains. This is a sexual differentiation of the brain, which is regulated by organizational action of steroid hormone. As I already mentioned in the previous slides, androgen action via steroid hormone receptor is very important for this sexual differentiation process. As I said before, since XX is a default type, XX individual be simply feminized to develop female type brain. On the other hand, in individual which has XY, both masculinization and defeminization occurs. In other words, not only it becomes more masculinized by the androgen action, but also default setting of female characteristics is disappearing in males. Once either female or male type brains are developed, steroid hormone act on specific brain site in social behavioral neural network to induce genomic action, which leads to expression of either female type or male type behavior. This is the activational action of steroid hormone, and the individual which has ovary secrete mainly estradiol, and individual which developed testes mainly secrete testosterone. These steroid hormones are transported to the brain and act specific brain site to regulate behavior. How these steroid hormones act in the brain to regulate behavior is the theme of the next section. Before to go into this detailed mechanism of action, I want to mention one more very important concept here. As I said before, during perinatal period, only testes produce testosterone, which is part of androgen. However, this amount of androgen and also even the way it affects on the steroid hormone receptors, which I will explain later, it's not dichotomy like we saw in the sexual differentiation. In other words, in this sexual differentiation process, degree of steroid hormone effect are continuously vary. Therefore, even final output of behavior, there are only female type behavior and male type behavior, which can be understood in continuous spectrum. In other words, there is XX individual which shows behavior very similar to male, or XY individual which is very similar to female. This is one of the very important concepts of today's lecture. Now I'd like to explain how testosterone and estradiol act on central nervous system. First, I'll talk about my studies in knockout mice. Before, I said steroid hormones are basically made from cholesterol, and as I showed here, testosterone, mainly secreted from testes, binds to its own receptor called androgen receptor after enters the neuronal cell. This receptor and testosterone complex move to the nucleus and cause genomic action, as I said before. Similarly, estradiol secreted from the ovary find its own receptor called estrogen receptor and cause the genomic action in various brain sites. One important thing is testosterone secreted from testes can be converted to estradiol with the action of enzyme called aromatase. This is called aromatization of testosterone. This occurs inside the brain. So, as I showed here, testosterone secreted from testes and taken up by neurons can be aromatized to estradiol intracellularly, and after that bind to estrogen receptors instead of androgen receptors and cause genomic action. To this date, there are two types of estrogen receptors are known, alpha and beta. Alpha and beta type of estrogen receptor are coded by different genes, but they are very similar in terms of ligand-bound area and also DNA-bound area. This means estradiol can bind to both alpha and beta with similar affinity. And also, receptor estradiol complex binds to very similar sequence of DNA to produce genomic action. Therefore, it's been very difficult to find the second type of receptor, estrogen receptor, ER beta, which only found in mid-1990s. I'll get back to the history of ER beta finding later in my talk. The fact that testosterone can be aromatize to estradiol and act on estrogen receptor means that even in males, estrogen receptor are very important for the regulation of social behavior. There are a number of 6 typical social behavior we are testing. Today, due to the time constraint, I will talk about mainly sex and aggression in males. This is a summary of my old study using estrogen receptor alpha or beta knockout mice. In ER alpha knockout mice, only estrogen receptor beta are expressed. On the other hand, estrogen receptor beta knockout mice, only estrogen receptor alpha are expressed. In ER alpha knockout mice, basically, sex-specific sexual behavior and aggressive behavior in male mice are both greatly reduced or completely abolished. Although I will not talk about today, but females' typical sexual behavior was also completely abolished in ER alpha knockout mice. On the other hand, not having ER beta in ER beta knockout mice did not affect, or at least did not reduce or abolish sex typical behavior such as male sexual, aggressive, and female sexual behavior. Sometimes we see prolonged receptivity in females or some degree of increased level of aggression in ER beta knockout mice. Based on these knockout mice studies, I conclude estradiol binding to estrogen receptor alpha determines on or off of expression of sex-typical social behavior, such as male sexual aggressive and female sexual behavior. On the other hand, estradiol binds to estrogen receptor beta, modulate levels or timing of social behavior expression. However, in knockout mice, either estrogen receptor alpha or beta are missing throughout their life and also entire parts of the body, including brain. So we don't know where exactly or when exactly estrogen binding to ER alpha or ER beta are important for certain social behavior. So rest of my talk, I will explain our recent findings to answer the following questions. 1. What is the exact role of estrogen receptor alpha and beta? And what is the site of action in the brain for the regulation of sex-typical social behavior? And finally, possible physiological mechanism of differential role of estrogen receptor alpha and beta. First, I'll talk about our study with estrogen receptor alpha knockdown at the specific brain site and specific time of development. In a few slides back, I told you that estradiol binds to both estrogen receptor alpha and beta with similar affinity. However, if we look at the distribution of ER alpha and ER beta in the brain, there are certain differences. This slide shows the overview of differential distribution of ER alpha, which is red, and ER beta, which is blue. In the ventromedial nucleus of hypothalamus, called VMN, ER alpha predominantly expressed. On the other hand, paraventricular nucleus, dorsal raphe nucleus, and locus coeruleus, in this area, ER beta are dominant. Finally, many of the limbic areas, such as medial amygdala, beta nucleus striae terminalis, lateral septum, and medial preoptic area of hypothalamus, express both estrogen receptor alpha and beta. In our first study, we investigate the effect of estrogen receptor alpha knockdown specifically in the either medial preoptic area, ventromedial nucleus of hypothalamus, or medial amygdala, and look at the effect on male sexual aggressive behavior. To achieve brain site-specific knockdown of ER alpha, we use adeno-associated virus-mediated RNA interference method. Here, complementary sequence for ER alpha is inserted in this AAV viral vector with marker sequence for GFP, green fluorescence protein. So by looking at GFP, we could identify where exactly this virus is injected and spread and taken up, and whether ER alpha expression is actually suppressed in those neurons. Here is some example when we injected this virus into medial amygdala. And here is a control, and here is knockdown virus injected animals section. These dots are all estrogen receptor alpha expressing cells. When we count number of ER alpha positive cells, there are clear difference between control group and estrogen receptor alpha knockdown group. Similarly, when we injected this virus to medial preoptic area, clear difference in ER alpha expression between control and knockdown group. Importantly, this knockdown is very site-specific since we did not see any decrease of estrogen receptor alpha positive cell in the area adjacent to medial amygdala or medial preoptic area here. Overall, 80 to 90% reduction of ER alpha positive cells in the target brain region. Similarly, in ventromedial nucleus of hypothalamus, we see very clear difference between control and estrogen receptor alpha knockdown group. And again, right next to ventromedial nucleus of hypothalamus, which is called arcuate nucleus here, we didn't see any decrease of estrogen receptor alpha in even knockdown group. In this bottom slide, the brown cells are the one express green fluorescence protein. That means the virus are actually taken up by these cells. In control group, also these cells has ER alpha protein, which is purple in this slide. On the other hand, the knockdown group, only the GFP is expressed and no ER alpha on these GFP positive cells. Okay, knowing that, we tested effect of estrogen receptor alpha knockdown in each of 3 different brain areas on 2 different behavior. First, we tested effect in the adulthood. So ER alpha expression is normal up to the point we injected AAV virus for ER alpha knockdown. And then few weeks later, we tested behavior. Please be aware that even though this letter is squashed, testosterone levels during perinatal period and also this huge increase during pubertal period is the same for both control and ER alpha knockdown group. So testosterone can be converted to estradiol, but it cannot bind to ER alpha in one of these brain areas. Okay, here is the result. In MEA, there is no effect on both sex and aggression. On the other hand, in MPOA, only sexual behavior was decreased, but no effect on aggressive behavior. Finally, in the VMN, both sex and aggressive behavior were greatly decreased. In the second group, we injected this virus in pre-pubertal period, which is about 3 weeks of age, and then tested sex and aggressive behavior in adulthood. In this case, we only tested MEA and MPOA, but not in VMN. Here is the result. In MPOA, the effect of pre-pubertal virus injection is exactly the same as this adult injection, which is only sexual behavior is decreased. On the other hand, and very interestingly, in medial amygdala MEA, pre-pubertal injection of virus greatly reduced both sexual and aggressive behavior, in contrast to no effect in adulthood. This means during pubertal period, this increased level of testosterone, probably converted to estradiol, binds to estrogen receptor alpha in the MEA and develop male-type neural substrate, which is necessary for the induction of sex and aggression in adulthood. Although I did not talk about sexually dimorphic nucleus in the brain during this talk, it is known that neuron numbers in the MEA is sexually dimorphic, which is male has a lot more neurons than females in this area. Interestingly, in our pre-pubertal ER alpha knockdown group, number of neurons in MEA is decreased compared to control group, almost similar level to female. Therefore, we conclude during pubertal period, steroid hormone, which specifically testosterone converted to estradiol, binds to estrogen receptor alpha in medial amygdala, further masculinize neural substrate for sexual and aggressive behavior through organizational action.